Hello, my name is Maximilian Stingl, and I will now present a workflow called OCMMED for obtaining cell-specific metabolic models through enumeration with DEXOM. This metabolic modeling method is based on constraint-based modeling, and more specifically on a technique called context-specific metabolic network extraction. Context-specific metabolic subnetworks are constructed with methods such as IMAT, which produce constraints for genome-scale metabolic networks from transcriptomics data. These constraints are then used to obtain a subnetwork which best fits the data by using an optimization algorithm which maximizes some objective function. However, these optimization problems are often strongly underdetermined, which means that for one set of constraints, there are in fact many different solutions which are all optimal according to the algorithm. Because of that, Rodriguez-Mir et al. Developed DEXOM, a package which contains algorithms for enumerating a large number of these optimal solutions, and thus obtaining a diverse set of context-specific subnetworks which all fit the transcriptomics data. Here, we present a Python-based workflow which uses these algorithms in order to reconstruct cell-specific metabolic networks. The main inputs for this workflow are the same as for most standard context-specific network reconstruction methods. You need one genome-scale metabolic network in a standard format, such as SBML, as well as some gene expression data obtained from one specific cell type or tissue, which can contain several different experimental conditions. The transcriptomics data is integrated into the network using gene-protein reaction rules. This step produces some reaction weights indicating which reactions the algorithm will try to activate in red on this diagram, and which reactions it will try to inactivate in green. You can choose between qualitative or quantitative weights depending on the data you have at your disposal, and it is also possible to provide these reaction weights directly without using the built-in GPR rules function. In addition to these main inputs, you can also provide some optional parameters such as custom flux bounds or forced flux through some reactions of interest. Then comes the main step of the algorithm, in which a predefined number of context-specific subnetworks are enumerated. In this toy example, 3 subnetworks are obtained, which all fit the transcriptomic data shown previously. There is then a step in which the results of this enumeration are compiled into one file, which we call the reaction activation frequency. Here, we simply look at each individual reaction from the network and count how many times this reaction is active in the enumeration solutions. On this diagram, the width of the reactions is proportional to the activation frequency, and the reactions with a frequency of 0 are grayed out. The final step of this workflow uses these activation frequencies in order to produce the metabolic model, and it contains 2 different options. The first option is a union network, which keeps all active reactions above a minimal frequency threshold. This option will produce a fairly large model that represents all the potential metabolic states of your experimental conditions. The second option is a network which only keeps the reactions with the highest activation frequencies and those which are necessary for passing flux through the network. This option will produce a much smaller model that represents the most important reactions or the core metabolism of your experimental conditions. Regarding the technical aspects of this method, because the enumeration of many optimal solutions is very demanding in terms of computational resources, we want to parallelize as many parts of this workflow as possible. In addition to this, we wanted to create a modular pipeline with several easily accessible options. Because of this, the entire workflow was written in SnakeMake, which is a Python-based workflow management system that allows users to create reproducible and scalable pipelines, and which is designed for being used on computer clusters. This diagram shows the graph of the different workflow modules for one toy example, in which we use 2 experimental conditions, and we perform 2 enumeration batches for each condition. In this example, we use an enumeration method called reaction enumeration to obtain a small number of solutions which are then used as starting points for the main diversity enumeration algorithm. After each enumeration step, the solutions are concatenated into a single file, and at the end of the workflow, the final module produces the cell-specific model. Finally, I will present a use case for this workflow. The workflow was developed as part of the European Goliath project, in which several biological models were exposed to different chemical contaminants. For this use case, we look at human hepatocytes and pancreatic beta cells, for which gene expression was measured with RNA microarrays by one of our partner labs. As part of this project, we want to produce cell-specific models that represent all potential metabolic states of our experiments onto which we can later map metabolomics and lipidomics data in order to explore the effects of the different chemical contaminants on these specific cell types. Because of this project design, for the final network option, we selected the union network with a low cutoff on the reaction activation frequency. As shown in this principal component analysis, the individual solutions of the subnetwork enumeration have a significant difference between the hepatocyte and the pancreas solutions. Due to the choice of the union network method, the final networks are quite large and have many common reactions, but still have some differences, for example, in the pathways for transport and drug metabolism. In conclusion, this workflow represents an accessible solution for cell or tissue-specific network reconstruction, which works on high-performance computer clusters, and which is quite adaptable due to having multiple options for both the input data and the output network. All the source code for this workflow can be found at the following link, and if you have any questions or comments, you can contact me using this email address, and both of these links can be found by flashing the QR code at the bottom left of the poster. Thank you for your attention.